Hi there. My name is Richard Condit, and I had a career as a university professor. I was a scientist, and I specialized in studying how viruses work. So I'm going to tell you how I came to that career and why a little bit, and what my day was like so that you could know how to do that. First, I want to point out that you and I have at least a little bit of a connection. I think my son-in-law, my daughter's husband, is Mr. Joseph Maines, who is the principal at Del Valley Elementary School. So you're in good hands. So without further ado, I'm going to share my screen here so that I can give you a little slide presentation as I go forward with this. And you have to be patient here for just a second because the program takes a little while to decide that it really is going to change slides for me. So this is my description of how to become a scientist in the context of being a university professor. So we're being patient here while this decides that it's actually going to change slides. Come on. There we go. So what does a university professor do? Well, in my case, and it's pretty typical at sort of the capacity that I did it in, my main job was to run a scientific research laboratory. Okay, so I had to set up the laboratory, get people to work in it myself, and do research in that laboratory. And this was research following my own interests. Also, in a university, one teaches, depending on the circumstances. That can be half the time, 3/4 of the time. In my case, working in a medical school is only about 1/4 of the time. And that means putting together lectures and being in a laboratory, being in a classroom and giving lectures. And also you help run the university. This gets called service. This means sitting on committees and stuff to help people organize how the university runs. And that's okay. So how did I become, or how could you become? By using me as an example, how could you become a university professor? Well, the first thing you really have to do is go to college. And that takes about 4 years. I studied biology. And that was the kind of science I was interested in. My father was a scientist, and I loved being outdoors as I was growing up. So I think I kind of inside me knew I was interested in biology and nature. And so I sort of pursued that while I was in college. I tried a bunch of different things, but settled on laboratory science. You come out of that with a bachelor's degree. That is a BA, a Bachelor of Arts, or an AB, Arts Bachelor, or sometimes a BS, Bachelor of Science. And in my case, I went to the University of California at Santa Cruz. I was raised in Santa Cruz, and this is a beautiful place on the coast overlooking the ocean. Then, if you want to pursue a career becoming a professor as a scientist, you go to graduate school. This means you go to another university, okay? And you're no longer just a student in the classroom. You do some classes, but mostly what you do is do a research project, an original research project in a professor's laboratory. This takes about 5 years. It can take a little long. In my case, it took 4.5. It can take as little as 4. It can take even less than that. Usually it takes a little longer than 5, but the average is probably 5 or 6 years. Importantly, they pay you, okay? You may accumulate some debt in college, but you won't accumulate as a result of schooling anyway, any more debt beyond that. They pay your tuition. You don't have to pay them. And they pay you a little bit of a stipend, enough to buy gas and groceries and maybe the occasional video game. When you're all done with that, you write up your research in what's called a thesis and or a dissertation, and you were awarded a Doctor of Philosophy degree, or a PhD. In my particular case, this was at Yale University, which is in Connecticut. So you can already see that I got an opportunity to travel around some. Then if you want to be a university professor, typically what you do is even more research and what's called a postdoc, or postdoctoral education. Once again, this is working in somebody else's laboratory. In a university or somewhere else. In my particular case, I worked at a research institution, and it's 100% doing a research project in somebody else's laboratory, so that by the time you're done with that, you really know how to do research. And importantly, once again, they pay you, and they pay you even more. They pay you enough to really, make some a halfway decent living, maybe buy a couple of video games. In my particular case, I did my postdoctoral research at a place called the Imperial Cancer Research Fund in London, England, and that was great fun. Once you've done all that, as I said, this for me was 3 years. I don't know if I mentioned that nowadays it can take a little longer than that. It can take 6 years, maybe 5 years, even longer. But if you apply yourself, you can get out before too long. And you're doing research. And for many people, it's the best time of their career. At that point, you're qualified to become a professor at university. So jobs are advertised, you apply for a job. And if you're lucky, they hire you. And in my particular case, I did 2 different universities. For the first 12 years of my career, I was at the State University in New York and Buffalo in a biochemistry department, which was in the medical school. And then I moved to Florida in 1990. And I was at the University of Florida in Gainesville, once again in medical school, this time in a microbiology department. And I was there for 25 years before retiring and then moving to Austin, Texas, where I could be closer to my daughter and her family. So what did I do during a typical day? About half my job was laboratory. And I'm going to say work in the laboratory, but I'm going to include other stuff under that topic. The thing I like to do more than anything else was design and do experiments. And in my particular case, I studied viruses, and I studied how viruses worked at the what we call the molecular level. That is the absolute minutiae. You've probably heard of DNA and maybe RNA and proteins and that kind of stuff. By studying viruses, you learn not only about viruses, but you learn how cells work at the very, very submicroscopic level. What genes are, how genes work, how cells divide, and et cetera. And so that was great fun. I would, as part of that, I got to advise students and postdoc and technical people in the laboratory, and you'll see some examples of that. In order to support the laboratory, I needed money, and usually you do that by writing an application called a grant to, say, a government agency asking for money to support your research. So that was a significant part of the job. And when I was done with experiments and had results, I would write scientific papers. That is a publication that's put in a scientific journal that other people can read, and they can read about your experiments. At least once, probably twice a year, I would also attend scientific conferences where a bunch of scientists get together and they present their research. So you get to present your research and you get to see other people's research. And for a guy like me, that was, like going to the movies all day long. It was really great fun and in great places. I got to travel all over Europe and the United States and some in Asia and South America. So there are great opportunities there. The other part of what I did during the day was to teach, and I taught college students and graduate students and medical students, and this means teaching in a classroom. That was about a quarter of my time. And another quarter of my time I spent on, as I said, service. That means sitting on committees and interacting with people to help organize your department and the university, and et cetera. You play a hand in making stuff run okay. So that's what I did during the day. Here's just some pictures of my laboratory now. I took these pictures as I was closing down the laboratory in anticipation of retirement. So they're not as sort of busy as they might have otherwise been. But anyway, you can see I had 2 big rooms with laboratory benches and shelving and all sorts of equipment. This is another picture in the same room, and you can see there's chemicals on a chemical shelf here and a couple of scales and a pH meter. Here's a refrigerator and a microwave and a fume hood and one of my colleagues working in the laboratory. Here is the picture from the other room. These 2 things are biological safety cabinets where you could work on cells and viruses and keep them sterile so that you were growing only cells and viruses and not bacteria and fungi, which would wreck the experiment. They also kept the viruses from getting out and about. Here's a couple of microscopes. Here's some water baths. So great fun. I love the laboratory. Here's a couple of people working in the laboratory. This guy here was a college student trying to figure out whether he wanted to do science for the rest of his life. And in fact, that's what he's doing. This was a postdoc at the time on her way to actually a job in industry. And remarkably, these two met in my laboratory and they're now married and have a kid, and they're both scientists. This is me doing my very favorite thing, which is doing science in my laboratory. And you can see here, I'm putting some stuff on a microscope slide here that I'm going to look at under a microscope and learn a little bit about how viruses work. And I worked in the laboratory all the way up until I retired. It was my favorite thing to do. Here is a micrograph, a microscope picture that came out of an experiment like that. And you can't see the cell boundaries here, but the blue bits, these are all done with fluorescent dyes and they're really pretty. The blue bits here are cell nuclei. So the cell in this particular case probably goes like this, surrounding the nucleus. Here's another cell around like this. And the green and red bits are bits of different parts of viruses that are growing in these cells. So by doing this type of experiment and changing the conditions and stuff, I could get information about how the virus was working in the cells. And I just love doing this stuff. It's great. So how could you become a science professor? Well, I've already kind of given you the path. The most important thing to do is go to college and pay attention while you're there. And by the way, I wasn't a terrific college student or even graduate student or anything like that. I was maybe a little above average, but the important thing was that I loved it. Okay, and it showed. Okay, and so people see that. They see that you love what you're doing, and they want to give you opportunities to do more. So, you know, as they say, find what you love and do that. And in college, one of the most important things to do is to find a laboratory to work in. And there are usually opportunities to do this, even if you're at a college that doesn't have research in the college. If you ask around with professors and stuff, they usually have associations with other universities where they can get you a position to do a little project in somebody's laboratory, or maybe you're interested in a different kind of science. You want to work in the field, study snakes or plants or something like that. There are opportunities to do that. Even in high school, there are opportunities to do stuff like that. You could probably find opportunities when you're in high school now to go spend some time in a laboratory, say at the University of Texas or something like that. So work in a laboratory, that gives you a feel for what it's like, and you get to find out whether this is something that you really like to do. And that's the most important thing. Is if you like doing it, okay, then keep doing it. I want to point out that there are alternatives to doing the kind of science that I did, alternatives to being a professor. Okay, following pretty much the same career path, college and then graduate school and then a postdoc. You could wind up working in a research lab. Recall I did my postdoc at a research institution called the Imperial Cancer Research Fund. Those people did nothing but research all the time. In fact, they didn't even have to write grants. Okay, it was funded by private funds. You could go to work in industry, and that could be a drug company, or it could be a biotech company that makes things for other people to do science with. And in fact, in industry, you don't necessarily even have to go to graduate school. You could enter an industrial path out of college, or maybe with an intermediate degree like a master's degree. And so the paths for those two, the research lab and industry, are really quite similar. You could also become a doctor and do medicine. And a lot of doctors actually do research in addition to being a doctor. And the difference there is that instead of going to graduate school, you go to medical school, but it's a similar sort of experience, except that if you're going to be a doctor, you work with patients a lot. So I retired about almost 7 years ago, and I moved to Austin to be closer to my daughter and her family, including Mr. Maines. But I've kept active during retirement. Among other things, for a long time, even before retirement, I participated as a co-host on a podcast about viruses called This Week in Virology. And this is a picture from actually before I retired, where we visited a high containment laboratory where they work on dangerous viruses. And we learned about how to work in a laboratory like that. So here we are dressed up in our biohazard spacesuits to work in that laboratory. And this is me, and this is my friend Vincent Racaniello and Alan Dove, both of whom worked on that show and on the podcast. So here's just some references. If you're interested in the podcast, it's called This Week in Virology, and there's a link to it. You might be even more interested. The American Society for Microbiology took our visit to the high containment laboratory and made it into a nice little YouTube video called MWV episode 68, Threading the Needle. TWIV goes inside a BSL-4, that's biological safety level 4 high containment laboratory. And there's a YouTube link there where you can watch the nice video they put together that says shows all about how what the laboratory is like, how you get dressed up to work in it, how you work in it, et cetera. It's kind of fun. And I want to point out that there's my email address, condit@ufl.edu, and you can write me anytime. And I'll try and tell you more, answer your questions, help you get on this path. So I'm going to stop share here for a minute because I got one more thing I want to show you. I want to show you my favorite experiment. So I'm going to put up this piece of paper in this dish. Okay. So this is called a plaque assay. It's a type of measurement that we do to measure viruses. And that's so you can see a Petri dish there. And I grew cells on the Petri dish. What you do is when it was when the cells were growing, wasn't purple. And it had a halfway up the Petri dish, there was a liquid that had food in it for the cells, made so they could grow right. And the cells sit down on the plastic. They stick to the plastic and they divide. And when they've divided enough, there will be a sheet of cells, a single cell layer thick that covers the bottom of the dish. So when you're done with the experiment, you can suck off the growth medium that's on the top, and then you can do what's called fixing the cells, treat them with formaldehyde, which freezes them in place and kills them and makes them, sort of glues them to the dish. And then I come in and also stain them purple. So that's why you see that purple covering on the bottom. That's a single layer of cells. If I were to look at that under a microscope, you could see the individual cells. But viruses are much smaller than cells. They actually grow by getting inside the cells and reproducing themselves inside the cells. So you can't see those with a regular microscope. You would need an electron microscope. So we use different methods to detect viruses, although I did some electron microscopy, and I showed you another microscope type experiment where I could get a look at what viruses did. But in this particular case, a regular microscope wouldn't do the trick. So what we would do is put a solution, a sample, onto the dish while the cells were still growing and had the red stuff on them that contained some viruses in it. And a single virus would then infect a cell on the dish and make more viruses and kill that cell and release the viruses to the surrounding cells. And they replicate on those cells as well and make more viruses and kill those cells. And again. And so what would happen is that where a single virus particle lands, it basically over time eats a hole in that layer of cells. And you can see those white spots. Those are places where the cells have died because a virus infected them initially and then grew and ate a hole. So from an experiment like this, I can tell that there are viruses there, and I can even count those holes and tell how many viruses were in the sample that I had. And I can tell other things about the virus as well by doing this kind of experiment. My very favorite type of experiment in the laboratory, called a plaque assay. So there you go. So I hope that's been informative. It's been great talking to you. I wish you the best of luck, and feel free to contact me if you need to. Bye.